Hi, I'm Ashley Leon, and I'm a first year in the Human Development and Family Sciences program. And this talk is co-sponsored by the Center on Aging and Population Sciences. And it is a great pleasure to introduce today's speaker, Dr. Grace Noppert from University of Michigan. So she also is affiliated with the Social Environment and Health Group at the Institute of Social Research. Dr. Noppert studies how social processes across the life course, such as socioeconomic disadvantages, may impact an individual's biological health. And more specifically, how the link between these social processes and different infections and immune function may help explain health disparities and advanced immune aging and the risk of aging-related disorders. So in today's talk, Dr. Noppert will orient us more to the biomarkers of immune aging that may be impacted by structural disadvantages and oppression. So that's really exciting. And at this time, I would like to remind you all that during the Q&A portion of today's talk, we would like to reserve the first two questions to graduate students. And with that, I would like to welcome Dr. Grace Noppert.
Thank you so much. So I don't need to do anything, just speak normally, right? They'll hear me okay? Yeah. Okay, cool. Well, thank you so much for having me. I'm so excited to be with you all today. I have to say it's a little like a kid in a candy shop because I'm looking out on so many scholars whose work I follow, who I respect as scholars in this field. So it's a real privilege and I'm very grateful for the opportunity. So today I'm going to be talking about the embodiment of the social environment and the architecture of the immune system. And like a good social epidemiologist, I have a colon here, implications for aging. And the reason I talk about implications for aging is I feel like if we understand this intersection, we actually can better understand inequities in the aging process. So I'm at the Institute for Social Research and I co-direct the Social and Environmental Equity and Infectious Disease Lab and the new Sociobiologic Lab, which I'll get to a little bit more at the end. Okay, so as far as an agenda, I'm going to start by talking through some foundational ideas. And then give a brief primer on the immune system as it relates to the work that I'm presenting, just so that we're all on the same page. And then talk about two different studies that we're doing, one that's published and one that's a work in progress, to look at immunosenescence at different levels of intersection with the social environment. And then ask some questions about where do we go from here, and then I'll take questions and close out. If there's a clarifying question in the middle, please do interrupt me. I don't want anybody to get hung up on something that I could just answer in the moment. So as a way of an introduction about me, I was trained in infectious disease and social epidemiology. In training in infectious disease, it really got me thinking, what do these chronic infections do to our body? And how does having an infection for a long period of time age your body, which got me into biomarkers of aging, and that was what I did my postdoctoral work. But for me, this intersection is really where the gold is, because I think there's opportunity to really think about disrupting health inequities. One of the things that we talk a lot about in my lab is that we actually don't need to document health inequities anymore. We need to think about how to disrupt them. We know that socially marginalized populations, disadvantaged populations have worse health because of the way that our society is set up. So we don't need to document that anymore. We need to think about and move towards disrupting that. So foundational idea number one. This is a paper that came out in 2018 from Tom McDade and Kathy Harris that's been really influential for me, because it lays out, so it's called the biosocial approach to human development, but it lays out this idea that these two domains, the social and the biological, are intersecting at multiple points across the life course and producing the health outcomes that we see in later life. So as a person that I spend most of my time looking at immune profiles of folks that are 65 plus, when I'm doing that, I'm thinking, okay, but actually this is a process that started a long time ago. That's the result of this intersection between the social and the biological. So on the top, they have laid out here the different domains of the social, from interpersonal relationships to neighborhoods and schools, all the way up to social structure and stratification. And on the bottom, they have the analog for the biological, which is really beautiful. Starting here at biochemical messengers, which are often what we call biomarkers, going to organs and tissues, physiologic and neurological symptoms in the genome and epigenome. And those things are intersecting across the life course in really critical ways that help us understand what's happening in later life. Foundational idea number two is this work on embodiment. So I'm going to quote Nancy Krieger, but there are a lot of folks doing really amazing work in embodiment. So she's not the only person doing it, but I like how she explains embodiment. She says that bodies tell stories about, and that cannot be studied divorced from the conditions of our existence, that often, but not always, match people's stated accounts, and that people cannot or will not tell either because they're unable, forbidden, or choose not to tell. This idea has been really foundational for me to understand this interplay between the social environment and biology, because this is the through line of my work, that if we can understand the stories that bodies are telling, then maybe we can start actually improving the lives of folks, right? So when I'm looking at biomarkers, what I need to be thinking at its core is that these are signals of a story that has been unfolding for a lifetime. So my job as a scientist is to learn how to listen to that story. It also harkens back to ideas in the biological sciences of priming and imprinting and sensitive periods, and it helps me bring these two domains of the biology and the social together. So why do I look at the immune system? It's a terribly complex system. It's really hard to understand. There are so many parts of it. Why would I look at this? So the immune system and the way the immune system ages is heavily tied to the social environment. I'm going to show you evidence of that today. And it's implicated in the etiology of many aging-related diseases and declines. And in fact, some of this work has been done with folks sitting in this room. Like the links to neurodegenerative disease and cognitive decline. There's a paper by Matteo right over here, a beautiful paper that talks about all the different links between inflammation and immune functioning with cognitive status. So we know that the immune system matters. There's also a really beautifully elegant epidemiologic paper on the link between Epstein-Barr virus and multiple sclerosis, causally linking those two, which has been a game changer in the last few years. I'm going to talk about where viruses fit into this story. But the immune system really matters for understanding aging-related processes largely. So the idea here is that if we can understand immune aging and the divergent pathways it can take across a life course, we may be able to better understand and address inequities in the aging process. So let's dig into the immune system a little bit. I will tell you, I am not an expert. I have taken immunology courses and don't understand most of what they say, but I really give it a God's honest try here. But the primer I'm going to give on the immune system is so that we can all understand the work that I'm going to show. So these are going to be basics. Okay, so this is the organs of the immune system. What it's missing here is the skin, which is a massive organ of the immune system. It's your first line of defense. But this is to show that your immune system is located. There are organs all over your body that are responsible for different components, different cellular components of your immune system. So some basics here. So the immune system evolutionarily was designed to combat threats, both internal threats like cancer and external threats like pathogens. It's also triggered by things like malnutrition and stress, which is relevant for a lot of the work that's being done by scholars in the room. Stress is a huge trigger for the immune system. There are two major domains that you'll hear about, the innate and adaptive immune systems, but the line between those is increasingly fuzzy. So we kind of artificially put certain cells in one category versus another, but really there's a lot of crosstalk between these two domains and a lot of cells that we talk about that belong both in the innate and the adaptive immune system. The best way to understand these kind of two components is in the language of specificity. So the innate immune system is a non-specific protective mechanism. So if you think about this schematic at the top here, so this schematic is going from non-specific, most general to very specific. So the innate immune system are things like inflammation. That's usually a whole body phenomenon. Your whole body is experiencing a hyperinflamed state. When you have the flu or an infection and you have a fever, non-specific, your whole body is experiencing that fever. Your body is raising its temperature to try to get rid of that pathogen. As you get more into the adaptive immune system, you get very specialized cells. These are very targeted cells. They're also very costly in terms of immunity. So it costs a lot for your immune system to develop these cells and to send them into a situation, but they're going to do the job in a much more specialized way. So this is where my lab spends a lot of our time thinking about the adaptive immune system. So in the adaptive immune system, it's responsible for the production of pathogen-specific antibodies. Those are your B cells. That's your humoral immune response. Sometimes you hear this word humoral and cellular. Humoral is your antibody response. My favorite cells in the body are T cells. These are part of your adaptive immunity. The antigens what you're having is your innate immune system is going to activate these different components of your adaptive immune system, including your B cells and T cells. And these cells store the memory of pathogenic invaders, which I will get to why that relates to the way that your immune system ages in a moment. Okay, so now my favorite thing to talk about are viruses here, and they're a huge part of the immune aging story. So persistent viral infections, many of us have these persistent viral infections, and for many years, they were thought of as a nuisance. So things like cytomegalovirus, Epstein-Barr virus, herpes simplex 1 and 2. For a long time, nobody thought that these were a big deal. In fact, when you got sick with them, most of the time you wouldn't even be sick enough to go to the doctor. But now what we're starting to understand is that these persistent viral infections, which you usually never clear, are actually aging your immune system in ways that are really important for understanding aging and decline and disease at large. So to give you, I will say this is our hypothesis, this is our hypothetical mechanism, because this is really hard to test. And we're trying to work out ways that we could test this. And if you have ideas, please let me know. But with these herpes viruses, usually what happens is early in the life course, you get a primary infection. So let's say you're a kid, you go to daycare. We all know daycares are hot spots for infections. You get infected with, let's say, cytomegalovirus. Your immune system dumps all these resources to try to get that virus to stop replicating and to push it into what's called a quiescent or a latent state. That process is really costly. So it keeps it into this latent state until something triggers it to reactivate, something like a stressful life experience or some other chronic condition or a disease occurs in your body. And all of a sudden this virus reactivates and your immune system is forced to dump again all of these resources to get that virus into a latent state. And what we think is that the frequency with which this reactivation and latency process happens is related to your social position in society. That folks that are more marginalized and disadvantaged are having this happen more frequently than people that are in more advantaged positions. So let me link this back to how this fits in with immunity. So let's say this is our pool of T cells. And these yellow cells are your naive T cells. These are your young, svelte cells. You're feeling great. Your immune system's super young. And you get this viral infection like CMV, and it forces some of your memory, some of your naive T cells to change to memory cells. Now, these memory cells are specific to that virus. So all of a sudden, your pool looks a little less young, and it has a few more memory cells floating around. That virus reactivates, and again, your pool now has to have more memory cells to come on board to deal with that virus. All of a sudden, your pool looks a lot less young and is full of a lot more immune cells that can't deal with any other infection except for that particular infection they have memory for. It's one of the reasons why among older people, we see less of a vaccine response for things like the flu vaccine. They have less naive cells to differentiate for that particular vaccine. So in this way, these persistent viral infections are accelerating the immune aging process. So immune control, when we look at one of the markers that we use is called CMV immunoglobulin G, IgG, it's your antibody response to CMV. When we look at that and we see high levels in an older person of CMV or even in a younger person, what that tells us is something about the overall fitness of your immune system and its ability to keep that virus into a quiescent state. Like I said, this process of latency and reactivation over the life course is affected by stress processes. And so then we use things like CMV IgG levels as an indirect measure of immune aging. And I'm going to show you, I'm going to use that in some studies I'm going to present today.
Just to clarify, is that CMV, is that general IgG, or when you put CMV, are you saying something, a subclass of IgG?
It's IgG specific to CMV.
Oh, I see. Okay, I understand.
Yep, it's specific antibodies to CMV.
It's IgG, but there's other IgG that's not specific to that.
There's other IgG that's specific to another pathogen.
Okay.
Yep, but when you test it, you would say, what's the Epstein-Barr IgG or CMV IgG? You're getting specific antibodies to a particular virus.
Thank you.
Yep.
And what is
Cytomegalovirus. So there's eight human herpes viruses. It's one of those. So it's a latent infection. Most people get it, and most people never clear it. So most of us in the room probably have it circulating in our body to some degree. So that's why it's actually an interesting virus to study, because it's very prevalent in the population, but what is differential is how well we control it. These are great questions though. Any others before I move on? Okay, so what sorts of patterns then do we see in immune biomarkers across the life course? We see an increase in inflammation. That's often called inflammaging. That's a lot of folks have studied this. I think that's really helpful. But now getting into the more cellular specificity, we see an accumulation of differentiated over naive cells. So we see that composition of your T cell pool. And actually you see the same things, although to a lesser degree in your B cell pool, in your natural killer cell pool, that you see this change from mostly naive to mostly differentiated cells that are differentiated to a particular virus or infection. We see a decrease in CD4 cells and an increase in CD8 cells, although there's a lot of debate about what's happening with CD8. So I maybe take that with a grain of salt. And we see less control of persistent viral infections like CMV. So you see what that looks like is actually more antibodies to CMV or other persistent viral infections in the blood. So now digging into the first study. So this is on population level differences in immunosenescence. Our aims here were to characterize markers and ratios of T cell immunosenescence in older adults, and then look at differences by age, sex, race, ethnicity, and education. This data is coming from the Health and Retirement Study, which is an ongoing nationally representative longitudinal survey of older adults, that is housed at the University of Michigan. It began in 1992 and includes over 20,000 adults over the age of 50. Follow-up occurs every 2 years, so still ongoing. It's a very rich data source. And importantly, they have what was called the venous blood study in 2016, which has been a game changer in thinking about immunosenescence. So for the first time, this was a major initiative, to say the first time that I know of, a major initiative in a population-based cohort to collect a wide array of biomarkers, more than just a few biomarkers of usually inflammation. Sometimes you'd get one or two other things. So this has been a really big initiative. So that's why we wanted to use those data to look at immunosenescence. So we got the immune function data from the 2016 VBS. We got demographic data from the 2016 core survey. We had 8,440 people with valid immune measures for the ones we were looking at. And we constructed 3 primary immunosenescence ratio measures. These have been studied before in other cohorts, although mostly really small cohorts. So this was the first time we were expanding into a population-based cohort. So we have the CD8, CD4 ratio. So this is just saying, okay, we know that there's a change from less CD8 and more CD8 and less CD4. So that ratio measure is capturing that. And then specific to both the CD4 and the CD8 compartment, we have ratios that are the effector memory to naive. So that's the most memory, they're your oldest CD4 cell compared to your youngest. And we have that in the CD4 and CD8 compartment. With all 3 of these measures, we coded it like this so that higher values is worse for immunity or more aged, however you want to interpret that, but higher is kind of worse. We also looked at CMV IgG antibodies. We looked at 4 categories of race: non-Hispanic, Black, Hispanic, other race, ethnicity, and non-Hispanic white. In sex, female or male, in age and years. In education, we had a 4 category here as well. Okay, so first we calculated descriptive statistics for the sample overall, and then stratified by age group, sex, race, ethnicity, and education, and then constructed a series of linear regression models with the outcome, one of those primary ratio measures that I just showed, and then the exposure with the sociodemographic characteristic. And these are the model building strategies. So model is what we would call our null model, because in this kind of work, you always adjust for age and sex. Model 2 adds in education, model 3 adds in race, and then model 4 adds in race and in education together, and then model 5 adds in CMV. I want to give a caveat here. One of the things that my lab really grapples with is how to control for race in a model, and what does that actually mean? And when we control for race ethnicity here, what we're actually trying to get at is race as a proxy for racism and the oppression experienced by those who identify as racialized minorities. So in this way, what we think is that racism is a confounder of both the education and the immune pathway. And so that's why we control for it. We've also looked at trying to stratify our models by race ethnicity group. In HRS, that's really hard because you have really small samples of non-white individuals. But this is an area that we spend a lot of time trying to grapple with how to do this well and thoughtfully, because racism is such a fundamental experience. It's affecting all of these pathways. Just adding a control for race ethnicity isn't going to be adequate, but that's what we have here. And anyways, if you have thoughts on how to analytically deal with this, I would be welcome to hear them, because it's something that we really, really grapple with. Okay, so demographics, the mean age of our sample was 67 years. It was 54% female, 10% non-Hispanic Black, 9% Hispanic, 78% non-Hispanic white. You see a good proportion of people with a high school education or less. And so digging into the distribution of these immune outcomes by sociodemographic characteristic. Just a quick note, higher, more aged immune profile. Keep that in the back of your head. Okay, so this is in the CD4 compartment. This is that ratio measure of effector memory to naive, so old to young. What you see in the red is the age group. So you see older age groups have higher values. That's what you would expect. You don't really see a sex difference. You do see a difference by race ethnicity. So you see that non-Hispanic Blacks and Hispanics had higher values than non-Hispanic whites and other. You do see a bit of an educational gradient, but where you really see that popping out is comparing less than high school education to the other educational categories. The CD8 compartment, really, I mean, there's just not much happening here. You do see an age gradient, which is oftentimes for us a sanity check. You would expect to see higher values in older ages. Not much else to report there. The CD8-CD4 ratio, you do see some interesting signals here. So again, you see the age gradient. You see a sex difference with males having higher values than females. Again, you see this race ethnicity difference comparing to non-Hispanic whites. And you see a really large difference here with less than high school compared to college plus. Like they're really much, much higher here. So what does this look like in the context of a regression? So this is the results for age. And so this is what the increase that you would expect with a 1 year increase in age. So model 1 is sex adjusted, model 2 is race, ethnicity, and education adjusted, and model 3 is CMV. So in model 2, you see that there's a little bit of attenuation when you add in race, ethnicity, and education, but not much. And then there's a huge attenuation when you add in CMV. I'm going to just give you the headline here. We don't know what to do with CMV. It's likely a complicated nonlinear process. And so do you model it as preceding these immune outcomes? Do you model it as a result of it? It's probably a little bit of both. So, that model 3 that CMV adjusted, we're kind of struggling with what to do with CMV. And I know, Matteo, you guys are kind of in the same boat of trying to figure out what to do with CMV in your models too. It's hard to figure out where to put it. This is in the CD8 compartment. You see a little bit of signal. Again, you see this attenuation in model 3 with the addition of CMV. And then in the CD8 compartment, you see an attenuation when you add in CMV all the way to the null. But you do see this increase in the CD8-CD4 ratio measure that's robust to controls for race, ethnicity, and education, which is model 2. So this is for age. For sex, what we see here, so this is in the CD4, this is the CD4 ratio measure. We see no real differences between the sexes, with males are the referent here for this ratio measure. But what you see is actually when you add in CMV, then you see that females have a lower level of the CD4 ratio measure compared to males. With the CD8 compartment, you see that across the board, females have a lower value of the CD8 ratio measure, and that gets even stronger when you add in CMV. And you see the same pattern in CD8, CD4. So you might be wondering, as I do, what the size of the coefficient, does that matter? And I'm going to just head this off at the pass. These are such new measures that we don't know actually clinically what they would actually translate to. So right now, this is like scratching the surface on seeing these differences, but then the next step is, what do they mean in context of decline in disease development? Okay, so these are the regression results for race ethnicity. So this is compared to non-Hispanic whites. On the bottom is Hispanics. In the middle are those that identify as non-Hispanic Black. And on the top is those classified as other race ethnicity. So in the CD4 compartment, we see a really interesting pattern where in model 1, we see Hispanics have a higher value of the ratio of measure compared to whites. It's almost completely attenuated when you adjust for education. And then when you adjust for CMV, it comes back. We see it happen a few times over. You see the similar pattern happening for those that identify as non-Hispanic Blacks. And you really don't see much of a signal as those classified as other, but it's a very tiny sample size. When you see the CD8 compartment, it's kind of all over the place. I'm not going to spend a ton of time because our results consistent with CD8 are just all over the place. In the CD8 to CD4 ratio measure, we see something really interesting happening. So we see here in Hispanics, they have a higher value than non-Hispanic whites on the CD8, CD4 ratio measure. It's attenuated when you add in education, and then attenuated to the null when you added CMV. And you see that kind of happening in all 3 race ethnicity groups compared to non-Hispanic whites. These are pretty noisy, as we might expect, but interesting differences what's happening with and without adjustment for education in the CMV. For education, so down here is less than high school education, and then we're comparing to college plus. So this is this, you can't see it with the Zoom thing, but this is the CD4 ratio measure. So what we see here is a pretty substantial difference. So those of less than high school education in model 1 had a higher value of this ratio measure compared to college plus. It's attenuated when you add in race ethnicity adjustments, then comes back when you add in CMV. What I think is most interesting to take from this is that you do see evidence of an educational gradient, in that as you get closer to college plus, the effect of education or the association with education gets weaker, which is what we would expect. But there's something really strong in the less than high school education group particularly. You see a similar sort of pattern, although you don't see this kind of funky finding when you add in CMV and it comes back with in the CD8 compartment. And then in the CD8, CD4 compartment, it's more what I would expect to find, where in less than high school education, they have a higher value of the CD8, CD4 ratio measure. It's attenuated when you add in race ethnicity adjustments, and then further attenuated when you add in CMV. And you do see evidence of an educational gradient starting to emerge. So what do these findings mean? They ask a lot more questions than I know what to do with right now, but I think one of the limitations of this work is it's a single shot in time, and it's still only a limited set of measures. So we really do need to move towards a system-based approach to studying immunosenescence that allows for feedbacks, that starts to capture this complexity more than what we were able to do here. We're only capturing this process cross-sectionally at one point in time when they're 65 plus or 55 plus, I forget what the VBS is. Immunosenescence is unfolding over the life course, and there's so much of this dynamic that we're not capturing at all. So we really need to move towards, at least having follow-up measures at the very least, and then probably, much more of a life course approach here. One of the big things in this area is measurement error in the measures that we use. And I think that should be something that we all talk about more. And especially when you're thinking about biological aging, there's a lot of measurement error in these laboratory measurements that's hard to capture and it's hard to get documentation on, but it should be taken into account when you're thinking about kind of the noise around these measures. And this is a huge issue for many studies, but especially HRS, is selection bias. That the folks that made it to be into the HRS are systematically different than the universe of individuals that we're trying to make conclusions about. And so I think selection bias is a huge issue that we need to deal with better in the HRS. If you have ideas on how to do that, please let me know. So the conclusions from this work, what we think is that this is preliminary evidence that the social environment is correlated with the extent of immunosenescence experienced by certain populations. Those occupying more disadvantaged societal positions, minoritized race ethnicity groups, lower education groups had indicators of advanced immunosenescence. But this is the real take-home message. What I think this paper adds to the field is that age has a long time been thought of as the primary driver of immunosenescence. And I think this turns it on its head. that we know that there is a larger role of the social environment than we previously thought. It's not just chronological age. So that's the real take home message here. Okay, so study 2. So that was at the individual level. In study 2, now we're trying to zoom out and look at neighborhood level associations. And do you see any signal between the neighborhood social environment and biomarkers of immunosenescence in the same sample? And do these associations persist net of individual level SES? This is a work in progress that my team is getting ready to write up. So we're still kind of grappling with the results. But I think there's some interesting associations that we see here, which is why I'm presenting it. But first, why neighborhoods? Why focus on neighborhoods? So neighborhoods are a really essential part of the social determinants of health. They're part of that larger constellation that I think we should really be paying attention to. And there's been a lot of work on neighborhoods, a lot of really amazing scholarship actually on neighborhoods and health, thinking about hard clinical endpoints like cardiovascular disease or Alzheimer's disease and premature mortality. But the question that asks me is, does the neighborhood also precipitate smaller, more subtle changes in biomarker measured health that might precede these larger disparities we see in these clinical endpoints? And so if we can kind of detect these smaller biomarker measured health, maybe then we can intervene on them as well. Studies from the Detroit Neighborhood Health Study have been really foundational in setting the stage for this. There's a study by Elizabeth McClure in 2019 that looked at increases in the proportion of abandoned homes in a neighborhood and associated that with decreases in thymic function, which is a measure of your naive T cell output. A study by Chantal Martin at UNC found that neighborhood quality and neighborhood disadvantage were associated with advanced epigenetic age patterns. All of this tells us there's likely a story here with how the neighborhood environment gets into cellular mechanisms of aging. But for me, thinking about the disruption of health inequities, neighborhoods are modifiable. You could think of interventions to actually improve neighborhoods for people that may have results for, in my case, the way that their immune system ages. And so that's why this is an area that I spend a lot of time thinking about neighborhood health, because I think that's a way that we can actually think about putting policies and funding towards improving neighborhoods for people. So again, we're using data from the HRS. What we did is we have their neighborhood environment data was based on the address of the respondent in 2016. We're going to start looking at where they lived before 2016 to start building kind of a life course. But to just, there is good evidence that folks don't move to many different types of neighborhoods over their life course. So where they're at in 2016 is probably a rough estimate of where they've been for quite some time. So we feel good about looking at neighborhoods in 2016. So the exposure here are neighborhood markers, and I want to, so I am co-PI of a study called NANDA, which is the National Neighborhood Data Archive. If you're at all interested in neighborhood research, check this out. What we try to do is curate publicly available neighborhood level data for a wide range of researchers, so that like when I used to do neighborhood work, I would create the measures on my own. And so then everybody's creating their own measure, and it's really hard to replicate science. And so what we have is this national archive of neighborhood measures that we theoretically vet. We do a lot of statistical vetting, and we're also always creating new measures. If you have an idea, get in touch with us. It's access through ICPSR. So we looked at two different indicators here: neighborhood disadvantage, which is the average of, and these are tracked, census tract level. So average of the proportion of female-headed households with children, proportion of households with public assistance income or food stamps, proportion of income below the federal poverty line, and proportion of unemployed populations. And then affluence is very distinct, and it is the average of the proportion of households with income greater than 75K, the proportion of population employed in a professional managerial occupation, and those with a bachelor's degree or higher. So they're composite indexes there. And so the value there ranges from 0 to 100. We looked at 4 outcomes of, oh, that got messed up. We got 4 outcomes of immunity. So the same 3 that I showed you before, plus CMV as its own thing now, its own outcome. And we standardize those measures so that we standardize and model the continuously. What you're looking at is standard deviations. And then we had a number of individual level control variables here. So the modeling strategy here is the immune aging outcome that we talked about, and then neighborhood disadvantage plus age and gender. And then we have a model building strategy that I won't go through all the way here. What we're looking at now is including Bonferroni corrections, because it's a lot of models that we're running. So that's on our radar as we're writing this up. Okay, so similar demographics as the study before. You can see the breakdown of the neighborhood variables and the mean variables, but I want to get into the meat here. So these are the results for the CD4 memory to naive ratio measure. And what we see here, so model 1 is adjusted for age and gender. Model 2 adds in education. Model 3 adds in race. And then model 4 adds in a number of controls, because we often get the question, what if you added in health behaviors, chronic conditions, all kinds of things. So we have model 4, but I think model 4 is over controlled. So disadvantage is on the left and affluence is on the right. So what we see is with each increase in neighborhood disadvantage, we see an increase in the ratio measure in the CD4 compartment. It gets attenuated in model 2, it gets attenuated to the null in model 3, and then model 4 is not terribly different than model 3 as far as effect estimates. With affluence, what you would expect is increases in affluence will result in declines in the ratio measure, and you do see that, although it's very imprecise and crosses the null. In the CD8 compartment, take-home message, you don't see a lot of anything. You see some signal of it going in the direction that we might expect, but the confidence intervals are very large here. The CD8 and CD4 ratio measure, we really don't see. I actually expected to see much more of a signal here, and we are trying to poke at this a lot as we write this up, because we don't see much of anything, and the estimates are pretty noisy. But where you see a huge story is with CMV. So these are continuous CMV antibodies, and what we see is that for each increase in neighborhood disadvantage, you have a 0.03 standard deviation increase in CMV antibodies. And I'm going to put that in context to make that a little bit more understandable about the effect size. And you see it attenuated, although it's robust even to model 4, which I think is very over-controlled. It's robust to all those adjustments. With neighborhood affluence on the right, you see it working in the direction that you would expect. An increase in affluence is related to decreases in CMV antibodies. So actually putting these in context now. That should say we see the strongest signal with CMV IgG, because I updated these results last night. But if I use model 3 for interpretation, a neighborhood that has a 10% higher score of neighborhood disadvantage is associated with a 0.12 standard deviation increase in CMV IgG antibodies. If you think about a neighborhood that's 50% more disadvantaged, that results in 0.6 standard deviation higher IgG antibodies. The thing that I want to frame here is that you're talking about a large structure like your neighborhood environment showing up at all in the way that your body's dealing with a persistent viral infection. That, I think, is where the need is. The estimates, I need to figure out what they mean in clinical context and what they mean for future disease development, and that's an area for future study. But the fact that we see this large social environment feature neighborhood disadvantage showing up in cellular aging parameters, that's where I think that there's something really interesting happening to the way that our bodies are dealing with the environments around us. So where do we go from here? This work asks a lot more questions than it answers, as is a lot of the work that I do, as it turns out. One of the things I really want to dig into is race, ethnic, and gender differences in these associations. When you're thinking about race, ethnicity, and gender, these are huge ways that our society is stratified, and they're seeping into every part of society. So how do we capture that in the models that we do? We're going to keep investigating the role of CMV and where it fits, and investigate mediating mechanisms. But kind of what I alluded to before, what does it mean that we see signatures of the neighborhood environment and cellular patterns of aging? Like, what do we do with that information? How does it help us understand health and aging? So I'm going to wrap up with some concluding thoughts, because I rarely get to be in a field with this many of friends and colleagues and scholars. And so these are kind of my thoughts on where this work is. So our work finds evidence that multiple levels of the social environment are embodied in the architecture of the immune system. I think what we're actually seeing is we're beginning to see the effects of systemic oppression in the process of racialization expressed on a cellular level. And actually work that's being done at ISR is looking at subcellular levels. I think our bodies are holding those stories, and we just need to learn how to listen to the signals that we have. We are just scratching the surface on these measures. And so I would say this about kind of the area of biological aging writ large. We're just scratching the surface. We're at the beginning of this, and this should be done with a lot of thought and caution. So as we do this work in biological aging, I think one of the most important things for us to consider is that it should be contextualized within the larger structures that put individuals in unequal contexts with unequal resources and unequal opportunities for health and well-being. I think putting the onus on individuals to change health behaviors without taking into account the larger context is dangerous. And equity needs to be centered, especially those of us that are doing this from the social or sociological framework. I think equity has to be centered in these conversations. And we need to acknowledge where we're at in this process and perhaps where we're not, especially thinking about kind of people latching on these ideas of accelerated biological aging. We need to be honest about where these measures are not and what they don't tell us. We have a new initiative at ISR, the Sociobiologic Lab, where we're trying to dig into where we're at with these different biological aging measures, these biomarkers of aging, and how do they help us center equity and bring everybody to the table and say, okay, we've got this measure CMV. What does it mean? What does it not mean? Where are we at in the arc of development? So stay tuned for more news about that, because we're pumped about this initiative, but it's hard to bring all these different scholars and thought partners to the table. And I have lots of amazing collaborators and funding support that I want to shout out. You can find me on Twitter, and that's where I follow all my immunology friends too. So if you're looking for more on immunology, get on Epi Twitter or immunology Twitter, and I'm happy to take questions.
Pressure's on the students. I know.
All right, first two questions are.
They do.
Yeah.
They do.
Yes. Yeah.
So you mentioned neighborhoods being modifiable and something that we can prevent and also invest in, whether policies or advocating, but how can we stop? There's like another notion of like neighborhood gentrification. So even if we do find findings that support disadvantaged communities, like I think there still will be some sort of pathway of like the low income people still being pushed out in that way. So is there anything
They're really good.
in the literature that has kind of talked about that, or what are your thoughts about gentrification when it comes to like, hey, we found, you know, investing in communities is helpful, but then it's still defeating the purpose because rich people or people with more money can move into these nicer, I guess, improved neighborhoods.
Yeah, it's a really, really good consideration. So I would say like intervening on the neighborhood for me is different than gentrification, where you're most often just having a lot of rich white people move in. And that's different, right? I think that there are interventions like improving access to healthy food or improving green space or getting rid of polluting sites that are disproportionately in communities of color. That is not the same as helping the people of color move out of a certain community. They're people of lower education and replacing them with advantaged, often white people that are higher educated. So I think we need to be really careful and really thoughtful when we think about policy level interventions for neighborhoods, that we're not promoting gentrification. We're actually promoting going into communities that already have community supports and actually saying, okay, what is it that would improve this community? Is it better sidewalks so that people can walk? Is it more healthcare facilities? One of the things we've seen in COVID is like, are there pharmacies for people to go get vaccines or go get medications? You shouldn't have to go 20 minutes away to go get medication. So it's a great, with all things, I think it needs to be really, really thoughtful. Yes.
I'm curious if you've looked at any of those physical or built environment variables that you just mentioned.
Yeah, actually, I have a really brilliant student, an undergrad, who's digging into these as far as like what makes a community really vulnerable to be a high burden COVID area. And he's looking at street connectivity and parks, and he's used the NANDA data that I talked about to actually look at all these different categories of businesses, like high congregation, high contact, low congregation, high contact, and to say, okay, which of these actually matter for what puts people at risk for an infectious disease? So we're actively doing this work, and we're creating new measures right now on police presence in neighborhoods and being able to say, okay, the census tract has this, per capita level of police presence, all these different features of the neighborhood that could matter, and trying to make these metrics at the census tract or zip code level to really look at these differences. So it's an area of ongoing investigation. Yes, ma'am.
So Grace, thank you so much for your talk, and it's a pleasure to have you here. So one thing that you mentioned that I want to ask you to talk a little bit more about is the measurement of race and the challenges that we have around race, particularly in biosocial research, right? So there's this kind of assumption that can happen that people run away with around biological difference. And we're always in biosocial research to try to push back against that. We need to have race in our models, but in terms of how, when you publish, because we need to have race in the model, how are you approaching in your manuscripts, talking about race explicitly so people understand the social construction of it and the intersection of that with the environment?
Yeah, it's a great question. And this has been an area where I've had to grow in the last 5 or 10 years. So let me preface it by saying I published a number of infectious disease papers early on, and I got people saying, oh, Black people have less of this thing, and that's why they have higher tuberculosis rates. And I thought I shouldn't have to be explicit about the social construction of race. Turns out I have to. And so what we've started doing is actually putting a paragraph in every one of our methods section that says, here's where we think about the social construction of race, giving citations to it, and being very explicit about how we see race in the models. And I still get people that read my papers and think that I'm talking about a biological mechanism for race. And so I don't know what else, like I struggle, like stratified models seem to be at least easier for people not to make that assumption. But then I get pushback from manuscript reviewers of, you're not powered to do stratified analyses and you have very wide confidence intervals and all this jazz. So it feels like, okay, it feels like an uphill battle, in one that is absolutely necessary, that I feel like we have to come out very explicitly and talk about race and what we mean. Because if we don't, I have had people misinterpret my work, and that's on me. I mean, do you have ideas?
I mean, I think that this is, and so I think that we should be having this conversation all the time, right? Because it's the same thing, in our work, we have to be explicit, the race is a social construction when we're going into like these kinds of larger settings. And so I think that at the outset, if you are addressing race in your papers and you're doing biosocial research, you have to be explicit about the social construction and making sure that that is of note. And maybe what we do is we put it up even further, not in the method section. We talk about it in the actual introduction to the paper. I mean, that's like, we have to just keep naming it in these interdisciplinary spaces, because people they can't get away from this whole biological difference in terms of race.
Even when they think they are, I find that they just default in these really subtle ways. Like, oh wait, that's like biological determinism about race, and that's not. I found with CMV, what I have been able to lean on are all the data on racialized minority populations being more exposed early on to CMV. And I can put that right up front and say, and this is why we think that race is important, not because of the biological and immunological thing, but because non-white people are being exposed because of neighborhoods, because of environments, to CMV earlier and more frequently. But yeah, you're getting at the heart of what we grapple with. Yes.
I just want to ask a follow-on question to that. See, this conversation, I hear it a lot, but what I don't hear about is how we ask about race. When we're active, I mean, a lot of times it's secondary data, and the data are 20 years old, but HRS is ongoing, it's collecting new data, and it still feels like, okay, it's 2 demographic questions at the end. Shouldn't we be asking it in a different way of people so that we get a richer response, a more multidimensional response to this being a social construct? I have no idea what the answer to that question is.
Yeah.
But it feels like it's a quick checkbox, and given all the conversation and worry and legitimate concern, it needs to be more than a quick checkbox.
I think so too. I mean, I think HRS asks people to self-identify, which honestly, coming from infectious disease literature is a huge step up, because an infectious disease surveillance study, and like think about COVID surveillance reports, that somebody who's interviewing you is assigning your race without asking you. So that for me is problematic. But what I think the HRS doesn't do well is there are huge categories that are missing, that are just clumped into this other race ethnicity. Like, what do we do about native and indigenous populations that are not reflected in this, that have very real, the way that we have racialized those populations have very real consequences for their health, and that's not captured at all. So I think it's an area for, I mean, I think we need to pay a lot more attention. And I think we're missing a lot. Like, I think even within, I mean, for example, if we pulled out the Asian group in race ethnicity, we're treating that as a monolith, which has wide implications for how they are treated in society, how they experience society. So I hear you. I think that we are not doing a great job in population health research on this, and it feels like we should be getting in on the ground floor of this. Yeah.
I'll stick with this theme, but in thinking about your neighborhood models, I was, you kind of didn't talk very much about why you chose the variables that you chose in your models, but it seems to me that one of the aspects of race that's happening in our country is really grounded in neighborhoods, right? Couldn't you actually do much more to be looking at that with your neighborhood models more specifically?
Are you talking about like thinking about structural, like segregation, the ways that,
All sorts of things.
so the way that we frame this work is to talk about that race is so critical to neighborhood selection and which neighborhoods you're allowed to live in historically and contemporary.
But also the attributes themselves and the associated schools.
Yeah, I think you're absolutely right. We don't delve into that in this paper because we're trying to get at this kind of crude association, but I think it's a good idea. I mean, one of the things I think about is, do we include segregation measures in this kind of work?
Exactly.
Do we include, we have a neighborhood school gap. So does your school match your surrounding neighborhood?
They're trying to make the point about social construction, right? Measure it.
Yeah, I mean, I've had the question of, should the percent Black or percent Hispanic be part of your neighborhood measure? I push back on that a little bit because what I don't want to do is assume that high proportion Black neighborhoods are disadvantaged. Now, you can make the argument that because of structural racism, that where we have allowed racial minority populations to live are disadvantaged. But I chose to deal with race in this way because the neighborhood measures with race included in that. I don't want to make that assumption, and so I chose to deal with it on an individual level. But this is the first of much work in this area that I need to dig into. It's a great question. Yes.
I would like to ask about your analytical approach there. So your story about the neighborhood effect, but you use the individual level regression models. So I wonder whether you have tried to employ some multi-level approaches more appropriate to think about the neighborhood effect.
Yeah, so this gets really tricky with this one. We're just talking about this. So the language is really nuanced here. So these are individual level, it's individual level neighborhood score. So we're not looking, I think that we could employ a neighborhood level effect, but it gets tricky. What my neighborhood researcher told me is that he said these are individual level covariates with your neighborhoods. And so we're not looking at neighborhood effects themselves. We're not looking at the larger neighborhood level. We're looking at the individual and what their neighborhood level exposure score was. But yeah, I mean, it gets neighborhood level regression analysis get really tricky to try to get the levels right. But yeah, in this way, it's all individual level. I don't think that we're committing the ecological fallacy in this one. Yes.
Can you clarify what you mean by when you say the neighborhood variable is at the individual level? So analytically, it is, but it's an aggregate measure of the movement.
Yes, but we're not estimating effects at that higher level. We're estimating effects at the individual level. So for individual X who lives in this neighborhood.
Yeah.
Yes.
You mentioned measurement error was the big issue with these particular biomarkers. Can you talk a little bit more about like what are the sources of that and what's being done to maybe get better measures or what?
Yeah. I'm also going to ping my colleague Lauren. So what I mean by that is like, if I would try to say this immune measure, this coefficient is equivalent to this many years of advanced immunological age. There's so much noise in making that, it could be plus or minus 10 years. And so trying to get those measures right. So there's laboratory measurement error, there's measurement in the batch. It's a whole, I mean, Lauren, you have anything? I mean, it's just a huge issue that we're all trying to get our heads around, and that is not my methodological expertise. I just know enough to hedge my bets of like, don't overextend this.
Yeah, I mean, I don't work with a lot of immune markers. My impression is that they're better than a lot of the other biomarkers that we work with that have even more measurement power, like epigenetic DNA methylation, where like if you run something on this chip and then you run it on this chip, I'm going to tell you your age is, 10 years different. So I think that like intentional partnership with our laboratories in terms of like how they're actually implementing the protocol and the measurement and making sure that we have like controls on every plate, repeats from the same person, so we can estimate like how much variation is there across the different batches. All of those things, I think the biomarker network is working really intentionally on, and a lot of other folks. So, but it's
I mean, it's like
challenge.
there's measurement error in like, where did you freeze and thaw it out? Did you get it from the same time at the same, there are loads of them. And so just trying to be honest that these measures are pretty noisy right now, and we don't have a good sense of them.
Okay. We're fighting. Fighting. Deb and I are fighting again. Grace, this was lovely. We're about to wrap up. Is there any more questions? Yes, one on the last one. The last.
I don't know how to pronounce this word. I talked to her about it. I
Uh-oh.
wonder if you're familiar with like autophagy or autophagy, and the
I can say
implication
autophagy.
of that in terms of immune system cleanup, recycling, and the fact that ancestral populations cycle through this kind of cellular cleanup, and when we look at like Hadza and other groups, we see very, very different age-related patterns of age-related function and all that. So I'm wondering, in terms of your interest in proactive solutions,
Yeah.
how do you think about those kinds of things?
I don't know a lot about autophagy. I like to say autophagy, but I'll get on Coulter, see if he says,
I
but,
say autophagy too.
does he say autophagy?
He has been hard, like I've heard it both ways.
Anyway, I don't know a ton about it. What I have read as far as immune cells is that sometimes what the dysregulation that we see in immune aging is that not enough autophagy is happening. And that has implications, especially thinking about brain health, is that there are senescent immune cells, like senescent T cells and B cells that are floating across the blood-brain barrier and causing neural inflammation, and that they're not being cleaned up. So that's the extent of my speculating in there, but I think it's a really interesting immune mechanism. I don't know how you would capture it well, like let's say a blood-based measure.
No, I think you need a research design that specifically capitalizes on that,
Yeah.
especially with the way we approach diet.
Yeah, I agree. Thank you guys so much.
Thank you.